Alright guys, we're going to get started with the biochem section, amino acids and peptides. All of the PowerPoints were uploaded late last night. So there's amino acids up there, there's nucleic acids, and there's lipids and fatty acids up there. And those will be the three things we're covering in the last 4 days of class. Also yesterday for lab, the last set of IR, NMR mass spec to be analyzed is up there. It's due on the 30th, which is Thursday, so make sure you get everything analyzed and sent back over to me, alright, by that time. So we're going to start with amino acids and peptides, which eventually will end up being our proteins. Okay. Alpha amino acids, alright, and our natural alpha amino acids here, as we said. So there's a couple things we need to look at. That there's 20 common alpha amino acids. You should be familiar with their structure. You should know their side chains. All of them have the same basic structure. Okay. So if we take proteins and we boil aqueous acid or base, yields an assortment of small molecules. So it's the way of breaking down our proteins, and we end up with these amino acids. The general chain is as you see here. Okay. There's some side chain. This is what changes in your amino acid. But every amino acid has this same structure. An amine end up here, the NH2, bonded to a carbon. Alright, bonded to a CO2H. Now this is known as the alpha carbon, and hence it's called an alpha amino acid. Okay, so you have your COOH, your acid part, you have your NH2, your amine part, all set up here for your amino acid. Every single amino acid or alpha amino acid has this general structure. Amine to a CH to COOH or CO2H as they have it here. This side chain changes, therefore changing the amino acid. So there's 20 different side chains for our alpha amino acids, all right, and determine what amino acid we have. Here are the amino acids. Okay. Now, there's 3 ways of naming your amino acids. All right, and this is a huge chart. It shows up better on the slides than it is on the video, but there's a full name. There's a 3-letter abbreviation, and then there's a 1-letter abbreviation. In all my time, I've only seen the 1-letter abbreviation used, okay, maybe a handful of times. It's usually the 3-letter abbreviation that goes with each that's used. So I would be familiar, if I were you, with not only the structure and the full name, but the 3-letter abbreviation. Okay, so what we're looking at is different side chains, and glycine is the simplest one. Okay. Glycine side chain is essentially a hydrogen. And then we start to build off that. Alanine becomes a CH3 group. Valine has a branch, leucine, and so on. Your goal is to know your amino acid via the side chain. That will pop up. You should know these, especially those of you who are going on to take biochem as your next course. They're going to have you memorize these, so you could be ahead of the game if you learn these now. Again, I don't waste your time having you memorize things that aren't going to be needed further down the line. Okay. Our amino acids then get broken down. They get broken down by nonpolar side chains versus polar side chains. So does our side chain have a charge? Doesn't it have a charge? What do we have? If I'm all carbon based and, for the most part, different carbon linkages, I'm probably going to be nonpolar. Okay. When I get into the polar side chains, they're going to have an oxygen. They're going to have something that's going to be a little bit more electronegative. Okay. A carbonyl carbon, possibly, like glutamine. Okay. Like asparagine, like threonine, cytosine. Cytosine was one that we looked at, or I looked at many years ago when I was in grad school, or it feels like many years ago, which it was, I guess. We were looking at linkages between cytosine and amino acid sequences. We can also break them down, besides having polar and nonpolar, we could also break them down by acidic side chains versus basic side chains. Aspartic acid, okay, and I'm going to tell you this right now. You're going to see the term aspartame, or you'll see aspartic acid, okay? The acid, it's the same thing. It's whether the hydrogen's attached or the hydrogen's not attached. If you're looking on the slides, actually where they write the name aspartic acid with the side chain, and glutamic acid should actually read glutamate and aspartamate, for the fact that they didn't put the hydrogen there. They have it as a charge. So if you're going to have it as a charge, it's not the acid. It's the acid when the hydrogen's attached. So technically, this is a little bit wrong on the table taken out of one of the books. We also have basic side chains. Those are going to be the ones that are amino rich, or amine rich, I should say, instead of amino, but amine rich. Okay. They're going to have an NH3, an NH2. They're going to have something in it. This again shows it with the hydrogen already attached. It shows it as a conjugate acid. Okay, and lysine, arginine, and histidine are those basic side chains. So yes, you should know what your side chains are and the name that corresponds with it. You should know whether it's polar or nonpolar, whether it's acidic or basic. So basically, you should understand this table. Okay. So let's talk about pH and pKa with our amino acids. So amino acids, as well as peptides and proteins, the reason that they're awesome is the fact that they have a acid end and they have a basic end. They incorporate both functional groups. Okay. The question is then, well, if they have an acid group and a basic group, and what about their side chain? What if, so they have an amine, they have the NH2, they all have a COOH, and then they have a side chain. What if their side chain's acidic or basic? How do we take that into account? How do we know what the pH of my amino acid when I place it into a solution? How am I aware of what that pH is going to be? Is it going to make my solution more acidic? Is it going to make it more basic? What's going to happen? Okay. So what we have to take into account is when I place it into solution, okay, what is the predominant molecular formula or molecular charge looking like? Am I more acidic? Am I more basic? And that's what's going to determine what the pH of the solution is. So it's all based on how does my amine end, how does my carboxylic end, how does my side chain look when I place it into an aqueous solution. So there is math involved. The Henderson-Hasselbalch equation, which has you be able to calculate pH given pKa, or be able to calculate pKa given the pH and the concentration of the acid, icebox, all that fun stuff is what's used. No, before you ask, you're not going to have to use it here. You're going to go take biochem, you're going to use it again. Okay? So, how do we determine the pH of something? Well, we do something called the isoelectric point. Okay? The isoelectric point is the pH of an aqueous solution of an amino acid, where the molecule has no net charge. Now, there could be an end that has a charge, but there's another end somewhere else that has the opposite charge. So let's say we place it into water. Our isoelectric point for the amino acid would be, let's say it's a nonpolar one, a nonpolar side chain. If I place it into water and the amine end becomes positive, then to have the isoelectric point, the carboxylic end must be negative, because there must be no net charge across the molecule. The negatives and positives must cancel each other out, and that's known as the isoelectric point. So here's alanine. Okay, alanine is our simplest amino acid. It has a side chain of just the hydrogen. Here we have a CH3, NH3 plus CO2H. Now here's where students get a little confused about this. If I am a base in an acid, I'm going to be charged because I'm going to be picking up protons. And we kind of talked about this in lab, how we got the aspirin to come out of solution at the beginning of the semester and all of that fun stuff. But if I'm a base, I'm going to be charged in an acid. Okay, at acid pH. Because there's going to be enough protons, enough hydronium around for me to pick up those protons. If I'm a base and I'm placed in a base, I'm going to remain uncharged, non-polar, no charge on me, because in a base, all my protons have been snatched up by the base. So my base N that I'm placing into that basic solution has no protons to gain. Similar. Okay. If I'm an acid and I'm placed in a base, I'll be charged, because in a base there's enough things available to take my proton. Or if we're going to talk in organic sense and not Gen Chem, we're talking Lewis, in a base there's enough electrons to be donated to my acid and take those hydrogens. Similar, if I, like a base, if I take an acid and place it in an acid, there are no electron donors available. They've been all used up, so I'm going to remain uncharged. So what they're showing here is, if I have a low pH for alanine, okay, and they're saying a pH that's less than 2, the amine end is positively charged. But there is no negative charge to cancel it out, so therefore there is no, and I'll go to the previous slide, isoelectric point. There's no pi. Because again, there has to be no net charge. Similar, if I go to a pH above 10, my acid end, my carboxylic end, the COOH end, will readily donate its protons and become negatively charged. But the amine end has no charge, and therefore my molecule overall has a net negative charge, no isoelectric point. Around pH 6, okay, around pH 6, I get both enough protons available for my base end to go and grab them, and enough base present to donate my protons and become negatively charged. So there's enough protons floating around for my base to pick up some and become positively charged. And there's also enough base at pH 6 around for my acid end to donate the protons. And we end up with an isoelectric point of 6.01. Well, how do I know what it is? Well, I could graph it, and I could do this in lab and measure the pH and so on and so forth, and that's what they did here. Or if I'm given the pKa 1, where I become an acid, and my base, or I'm acidic enough for my base to be charged, or my pKa where my acid end donated its protons. If I take these two pKa's and average them, I get the isoelectric point. 9.69 plus the 2.34 is going to be roughly your 6.01. So our key to find our isoelectric point is to have our charges equal out across the molecule. So let's look at some different molecules here. Okay. Here's arginine. Okay. Aspartic acid, isoleucine, alanine again. Not only in some of these, and that's why we're showing these here, not only in some of these do we have to worry about the amine group that's always there and the carboxylic group that's always there, we also have to worry about the side chain. Now my side chain in alanine is just a hydrogen. My side chain in isoleucine are just carbon. So we're really just balancing the amine and the carboxylic acid out and getting them charged up. Over here with arginine, I've got two possible amine groups, here and here, and I got a carboxylic acid group there. In aspartic acid, I've got two carboxylic acid groups and one amine. Now, I'll point this out. If I have additional amine groups on my side chain, my pi is going to be a high number. If I have additional carboxylic acid groups, okay, so glutamate or aspartamate as we're looking here, then my pi is going to be a low value. If I have nonpolar side chains, we're going to be roughly in this middle 6 value. So, nonpolar side chains, hydrogen, all right, our carbon chain down here, pi is around 6. Side chain that contains another amine, my pi is going to be a higher number. Side chains that are a carboxylic acid, my pi is going to be a lower number. All right, any questions on what we covered so far? So you should know your amino acids. You should know their names and their side chain. You should know whether the chain is a nonpolar, a polar, an acidic, or a basic. If I were asking this on the exam, I would ask it two different ways. I'd either give you two pKa's, all right, for the, I'd say, okay, here's the pKa for when the acid is, you know, the carboxylic acid end is a minus and the amine end is a positive. Okay, so that's one thing I would do. Or I would ask you a question like this. I would put like arginine in there and say, what do you expect the pi value to be? Do you expect it to be 6? Like, which of the following would be an appropriate pi for arginine? 6, 7, 3, or 10? Okay, so I'd either have you calculate it, and all to do to calculate is take the two pKa values and average them, or I would ask you a question like that. Hey, here's alanine. What would you expect the pi value to be? 10, 2, 6, or 11? Okay. Now, this is where we get into testing for the amino acids and everything. So there's an organic reaction that you may see on CSI and things like that, known as, they'll walk in, they'll be looking for amino acids, or they'll be looking for evidence. They say DNA evidence usually, which is wrong. But they'll spray this stuff on there and it'll turn purple. Okay. You touching things, you speaking, you doing, especially nowadays, like we're well aware of this after all of this, but you touching things is leaving behind evidence that you were there. You just being in a place is leaving evidence that you're there. You're shedding skin cells there. I mean, I know the one person up at Keystone was studying at one point, extracting DNA from skin cells, extracting like small pieces of genetic material from skin cells. We're leaving behind our residues everywhere. Well, one test you could test for is, you touch something, you're going to leave some of your amino acids behind. And it's known as the ninhydrin reaction, and it actually will turn purple. Okay, and the thing about ninhydrin is ninhydrin's really toxic. Okay, ninhydrin's a very toxic compound. If we were using it in a lab, we'd have to wear a face mask. Even in the hood, it's necessary to wear a face mask. You wear, double gloves. Okay, I mean, in an academic setting, I can't tell you about a professional setting, but in an academic setting with liability and all of that, we would take all the precautions, all right, of using ninhydrin. So amino acids interact with this ninhydrin. Okay, this is known as, this is the full name of ninhydrin, triketohydrinidine hydrate, okay, known as ninhydrin, and will create a purple complex in the presence of amino acids. Okay. So here is the general reaction for an amino acid reacting with ninhydrin to give us the purple color. Okay, so our amino acid is represented there as the second thing with the NCH3CO2. All right. And we have an interaction, all right, that occurs. The product of the interaction of ninhydrin and our amino acid creates this compound right here. Okay. This compound, and I'm going to show you exactly where here. Okay. Let me get my pen. Or let me go back. Pen, red. That right there, that linkage. A C that's bonded to R groups. So this C right here. Okay, that carbon right there. Double bonded to an N that's bonded to another carbon right there. This is known as a Schiff base. Now again, those of you who have to take biochem, you're going to see Schiff bases brought up again. I actually know one student I had last semester, I was helping with biochem, and she got all excited. She sent me a message and said they asked the class, does anybody know what a Schiff base is? And I raised my hand. And the teacher was surprised that one of us knew what a Schiff base was. And she said, I learned it in organic. So a Schiff base is a carbon, okay, that's bonded to other carbons that's double bonded to nitrogen. So it's a C double bonded to an N that's bonded to two other carbon chains. That double bonded N is then bonded to another carbon. So that's why it says a destructive to the amino acid. So here's the deal with this. If I'm going to test for the amino acid, I'm going to destroy the amino acid. So I'm not going to be able to tell what amino acid is. Basically, what this test is for is a positive test for amino acids. It's not going to isolate them. It's not going to do anything with them where you can basically look and say, I have this amino acid. No, the amino acid is going to be destroyed. So step one is we're going to create a shift base. And then through another series of steps and another addition of ninhydrin, we create a compound that looks like this, which could also be considered a shift base. I've got a C, it's double bonded to an N. That's bonded to a carbon chain. Here's my shift base. Here and here. Carbon, double bonded to an N, bonded there. Am I going to ask you this mechanism? No. Should you know that ninhydrin is a test for amino acids? Yes. Should you know a positive test is purple? Yes. Should you know what a shift base is? Yes. Should you know that the amino acid is destroyed when testing for it? Yes. But am I going to ask you the mechanism or the exact reaction? No. The shift base is named after Hugo Shift. And again, here is the general form for a shift base. These could be hydrogens. They don't have to be R groups. This has to be an R group. It's a carbon double bonded to a nitrogen bonded to a carbon. That's really the shift base part. These could be two hydrogens. These could be two carbon chains, a carbon chain and a hydrogen like they show here. But this 100% has to be a carbon. All right, so let's talk about another reaction. All right, electrophilic halogenation. Electrophilic aromatic halogenation. All right, we've talked about this before. It's EAS reactions. Okay, and it's going to be important for what we're talking about today. So we're going back down memory lane to EAS reactions. All right, we take a benzene ring, we take our halogen, we take our catalyst, whatever it is, H2SO4, whatever we're going to use as our catalyst, and we get our halogen to add to our benzene ring, and we get fluoryl chloride or chloral or bromo, all right, benzene. So bromobenzene, chlorobenzene, fluorobenzene, iodobenzene, whatever it is, it's probably going to be chloral or bromo, as we talked about the reactivity of iodine and fluorine. So just to review with you, our catalyst to do a halogenation along with our H2SO4 to get it going has to be a metal halide, whether it's iron bromide or aluminum chloride, to get our bromine to add to the chain. Okay. Just a little bit of review of EAS. So, to show that halogens can move and things like that, and look at electrophilic aromatic substitution and all this fun stuff. Well, how were amino acids synthesized? Where did they come from? Well, we can make them in a lab, but we and animals with amino acids and so on were around before we could make them in a lab. So what is our things here? All right. How are we going to make amino acids? Okay. Amination of alpha bromo carboxylic acids shows a way of making our amino acids as we know them. Okay. So here's method number one. We are going to take ammonia. We are going to place it in a reaction, okay, with a bromo-substituted carboxylic acid. Okay, you need a halogen-substituted carboxylic acid in the presence of ammonia. Now, I had a student at Keystone who's finishing his master's now, who basically made pre-Earth conditions. And there's an idea out there that the rocking of the waves in pre-Earth conditions is what brought the amino acids together to start forming peptides and proteins and things like that. Okay. The thing about that is this. It actually worked. He's actually forming small peptide bonds. And we'll talk about peptide bonds in a little bit. They're reactions of amino acids to make our proteins. So this is looking at a way of how could amino acids been made. Well, what we're going to do is a substitution reaction, an SN2 reaction, a one-step SN2 backside attack with a really good leaving group. We react our bromo-substituted carboxylic acid, and it would have to be a 2-bromo, because here's 1, here's 2. We would need it to add here. So a 2-bromo something anoic acid with ammonia. By doing so, the ammonia group adds substitutes where the bromine was. And if you see really close here, and look, now I've got the COOH end, I've got the amine end, and I've got my carbon, where R is going to be my side chain of what my amino acid is. So we call this the amination via ammonia amination of a carboxylic acid or bromo-substituted carboxylic acid. Okay. So that's one way of making a amino acid. Number 2 is Gabriel synthesis. Okay. What's great about Gabriel synthesis is that you don't have to start with just a carboxylic acid. You could actually start with any bromo with a COOH. So like an ester will work also. Here is the reaction. Again, I'm not going to ask you for the mechanism of this reaction. But you should know that you need potassium phthalimide to start a Gabriel synthesis. Okay. You should know that there's an acidic hydrogen that's available to be plucked off. And then we end up with an amino acid. Again, I'm not going to ask you Gabriel synthesis step by step, but you should know that it's a method of creating an amino acid. You should also know that it allows not only using carboxylic acids to start, but also esters. Okay. So you should know what the steps or what's going on in the Gabriel reaction, not necessarily step by step by step. Okay. To me, this one, besides the just, hey, let's just throw an amine at the bromo, I think the Strecker synthesis is the coolest way, the best way, not the best, but the neatest way, the cleanest, that's the word I'm looking for. The cleanest way of creating an amino acid. Besides, hey, let's just dump a whole bunch of ammonia in. It allows us to start with an aldehyde. It also allows us to start with ammonia and the aldehyde to make our compound. So, the first one we looked at was a carboxylic acid. The second one, Gabriel synthesis carboxylic acid or ester. Here, we're going to start with a carbonyl carbon, and we're going to start with an aldehyde. You react the aldehyde with ammonia. Okay. And you end up with this product here. Do we remember what that product, this product right here, is called? What is the product of the first step of Strecker synthesis? You got it, Jen. That's an amine. Now, let's ask you this then. If this hydrogen was a carbon, it's also an amine, but it would be known as, more specifically, what we talked about, a Schiff base. So by having it here, so a Schiff base is really an amine with a specific name and a specific look. But you're 100% right. It's a carbon that's double bonded to a nitrogen that's bonded to a functional group. It's an amine. So right here, adding to what we already know, right, this is an amine. We'll see how well I can write here with my mouse. Perfect. Now, what they don't put on here, and they should, they kind of forget to put it here. That was awful. I'll write it above here. What they forget to add is that we're going to add some sort of cyanide. Hydrogen cyanide, something, the cyanide anion, sodium cyanide, some sort of cyanide anion, to essentially get rid of that double bond and end up with this product right here. Okay. And then we can oxidize the cyanide side chain. Okay, oxidize it with water in an acid or a base. Okay, form, and that cyanide will become CO2, and you end up with your amino acid. So to me, this is a little bit cleaner. I understand that the first method, which is the simplest, but this is a little bit, Strecker synthesis is a little bit cleaner than Gabriel synthesis, to the fact where you could essentially sit here and say, boom, I'm going to add ammonia to make an amine. I'm going to take my amine and we're going to reduce it because we're going to we're going to oxidize it because we're going to remove hydrogen or a double bond, all right, and create the cyanide. All right, so we're going to lose electrons from the nitrogen. The nitrogen's going to lose electrons and give them over here. All right. And then we're going to oxidize the cyanide and turn it into a CO2 and end up with that same functional group. All right, or same basic structure, I should say, with the functional group. Here's our functional group and our same basic structure of CH with a CO2 and an NH3. Okay. So that's 3 ways, all right, to create a amino acid. Here's the breakdown of everything happening. The ammonia attacking the aldehyde. So, carboxylic acid with bromine, a bromocarboxylic acid, one way to start. A carboxylic acid or an ester, that's Gabriel synthesis. An aldehyde can get us there. So an aldehyde in a very electron or a very ammonia-rich environment could end up making, amino acids. So could that have been one way amino acids started forming? Okay. Possibly, but then you would need the cyanide richness and things like that. It's more likely that there was halogens available. Okay, and we had a carboxylic acid would have substituted halogen, and in an ammonia-rich environment, we just started making small amino acids. Okay. And this is showing the rest of the steps along the way. We don't really have to worry about any of that, but where I want to go with this, since I already ended the thing, is, well, what do we do with these amino acids and why is it important? Well, the key that's important to our amino acids is the fact that once we have our amino acids, and again, a lot of factors go into play here. So we have our amino acid site, our basic amino acid here, okay? And we have something that looks like this. Okay, and we have some functional group on it. We have another amino acid that's hanging out over here. Okay. Like so. With another side chain. It could be the same side chain, it could be different. Now, when they interact with one another, you have a base end here, you have an acidic end over here. Okay. Now, in biology, they're going to call it the N-terminus and the C-terminus. Okay. This is the alpha carbon. The side chain is off the alpha. Okay. And we have the N-terminus and C-terminus, which we know as the amine N, all right, and the carboxylic N. Now, what's important here is, and I'm going to take this NH2, okay, and I'm going to take this COOH, and I'm going to draw them out like a Lewis structure a little bit here. Okay. And this here like this. What happens when we make a peptide bond? Okay. Is that the OH from my acidic end, my C terminus, and the H from my amine. Okay. Drive off water. Water is removed. It's actually a dehydration reaction. We end up with a linkage. And right here is our peptide bond. Now, we'll continue to do that, and these side chains are important. We don't want them to be, big bulky ones next to each other due to steric hindrance. There is a gap there. You don't want, charges could dissociate. And what gets crazier and crazier as I add more and more peptide bonds, these side groups can interact with one another if they're in close proximity. Proline, when you look at that, there's something known as the proline hairpin turn. Cysteine, and this is what we were looking at. Cysteines could interact with one another, both different molecules and within each other, if they're close enough, and they can make sulfide bonds with one another, which could cause your amino acid chain to defunk, all right, or stop working properly, all right, the protein that it's signaling for. So all of this, don't forget, is taking place All right, we're taking these amino acids and we're making our peptide bonds and we're turning them into proteins inside the cell, inside the rough ER, specifically in the ribosomes. So the biochem behind this is amazing. Because in a water-rich environment, the water is going to hydrolyze this and break that peptide bond. So we are in a water environment, but we got to do it in such a way where we're being protected. So you're going to have nonpolar and polar, and the way the protein folds, the nonpolar has to go on the outside, and the polar amino acids have to go to, the polar amino acids go to the outside, the nonpolar go to the inside. So how I'm linking them is very important, not only to the function, but how it folds. So your protein ends up being a piece of, a long chain of amino acids. That's known as your primary structure. And then it starts to fold, and you'll learn that there's 4 structures. There's primary, there's a secondary structure, which talks about how we're turning and twisting alpha helixes and beta sheets. And then you talk about the specific folding in your tertiary and quaternary structures. But essentially, you have your amino acid chain here as this flat piece of paper, and then it starts to fold. Okay, and when it's done folding into whatever its functional, form is, on the outside is going to be your polar amino acids, because this is all water out here. And on your inside is going to be your nonpolar amino acids. Okay, because they're going to be hydrophobic and they're going to go to the inside. What happens in an oxidative state, or again, if our amino acid sequence is off, or, just the normal oxidative state of aging, these proteins could start unfolding. Well, if these proteins start unfolding, then the hydrophobic ends are now exposed to water, and they don't want to be exposed to water. Somewhere else, there's another protein that might also be unfolding. And now I have 2 hydrophobic ends exposed to the water environment that doesn't want to be there. And what they'll do is find each other and bond up to each other. And now I took 2 functional proteins that were working and doing some sort of job. And now I've got one big mess that's doing absolutely nothing and a wasted group of peptide bonds. So it's very important, the not only order and what peptide bonds, but also when those peptide bonds form and we start forming our proteins, do they fold properly? Because if they don't fold properly, you're going to get a non-functioning protein sitting there. You're going to be getting something that has hydrophobic ends exposed. So what do they tell us to do to prevent aging? Again, have antioxidants and things like that. Does it work? I don't know. Some people say yes, some people say no. Some people say the amount of antioxidants you'd have to eat or ingest to have any effect isn't humanly possible. Some say, well, a little is better than a lot. So, the jury's out there. Unfortunately, we don't have a choice in the aging matter. We're all going to age. But it is important to know that it is a dehydration reaction. We are linking the acidic end with the basic end. We are creating water in the process. And then what amino acids I am is going to determine where I am in the protein folding. Okay. So that pretty much ends amino acids. I'll have your exams graded. I'm working on them. I had lab stuff I had to get through this weekend and all last week for my other courses. I will get those grades up by Friday, I promise. Okay.